Hi, everyone. Welcome to the Physionic Podcast. If you're new here, my name is Nicholas Verhoeven. I'm a PhD student in molecular medicine, and today I've got a really interesting topic based off of a study is it a study? I guess it's a case study that's hot off the press as of less than a week ago. So I'm going to dive into and answer the question of, can we be reinfected with the SARS-CoV-2 virus, which leads to COVID-19? So what we've been struggling with over the last, since, I suppose in particular areas of the world, since end of last year, and in the United States, especially in March, from March onward. So I'll be going through the background, going through the results and what we can take away from this study. And there's several different key takeaways that we can get from this paper. So I'm really excited to get into that. But first, I'd like to let everybody on the podcast know, if you haven't seen my announcement on YouTube, over the last several months, I've been writing a book, and that book is now finished in terms of the actual writing process. It is a book called, at least so far, it's a working title. It may end up changing, but most likely it'll be something similar. So it's called Understanding Creatine, and I'm currently in the works of actually formatting it to actually make it look nice and professional and whatnot. It'll be an ebook. It is going to be updated regularly based off of the research that comes out that I come across. And it'll be updated without any charge to anybody who ends up buying the book. So once it's formatted, then I'll actually release it and actually have a link for you to get it if you care to check it out. Also, I'm going to have a video course that goes along with the book. You can get the video course with it, or just get the book itself. The book has over 120 scientific references, and I've been working on it almost every single day. So it's been certainly a labor of love over the last 3 or 4 months. And I cover a ton of different topics. I actually have a specific chapter for women to distinguish the differences for women when it comes to creatine. I talk about creatine supplementation, of course. I talk about not just the effects that creatine has on muscle, but I have the effects that it has in the brain, the effects that it has in different pathologies, if that's like diabetes, if that's Huntington's disease, if that's any number of different things, muscular dystrophy. So it really covers all kinds of different things. And there's certainly information that's going to be in this book that you're probably not going to find really anywhere else, because some of the things are more nuanced. It's not like there's a study that specifically probes a particular question, but after reading, over 100 studies, you get to a point where you're like, I'm starting to see a trend here. So anyway, I did find a few trends that might be interesting, and I think that they'll be definitely unique, things that other people probably haven't considered when it comes to creatine supplementation, the type of creatine, things like that, and maybe who should be avoiding creatine as well. So anyway, that's where I'll leave that, but I just wanted to update the podcast that the written section is finished, and I'll let everybody know once it's actually up and available for purchase. All right. With that, if you want to look at the data, I have a little bit of data when it comes to this particular paper. There's not much, but I have the gene sequence that I end up discussing a little bit. So I'll be discussing that on Instagram. So if you want to actually look at that, as well as some more details that's related to this particular study on reinfection from COVID-19 and what it means, then hop on over to Instagram, where you'll get a more detailed breakdown. All right, let's jump into it. Now, basically, like I mentioned earlier, this is a case study, meaning that it is a study of a single person, just one person. So we're not looking at averages. We're not looking at really a bunch of numbers and stuff like that. We're not looking at bar graphs. They're just seeing and testing this one individual. So this person was actually infected with COVID-19. Again, this is really technically SARS-CoV-2 is the virus itself, but this person was infected with SARS-CoV-2 and was infected back in March, and then was released from the hospital. So it's a man, 33 years old, was first infected on March 26th and then discharged clear of the virus. So they tested him multiple times on April 14th. So they tested him with he had to have 2 negative tests, and he came back with 2 negative tests on subsequent days. So he was released on April 14th. So had COVID-19, got over it, and left the hospital. He is 33 years old, in good health, just presented with the typical symptoms: cough, sore throat, fever, headache for 3 days on that first infection, and then ended up recovering. Then he was in Spain, and when returning from Spain, so this is on sometime in August, late August, he was returning from Spain and tested positive on re-entry screening to China. So he was at the hospital, and to enter China, apparently, you need to go through screening, and they ended up finding out that he tested positive. So that must have been quite a shock to him. But he was hospitalized and had a normal temperature and no symptoms, but one inflammatory marker was elevated, which is C-reactive protein, which is kind of a general inflammatory marker. But if you look at some of the other data, looking at some of the immune cells, like the amount of immune cells, white blood cells, neutrophils, lymphocytes, things like that, those were all in the normal range. Like they weren't going way off the chart or anything like that, but his C-reactive protein was elevated and his potassium was a little low, but otherwise perfectly healthy, even though he tested positive for the virus. And it's true. I mean, if you look at the data, again, you don't have to look at the data. I'm essentially explaining everything that you need to know. They tested for the viral load, so the amount of virus that is in his system, and sure enough, he did have high levels of the virus, again, after he had tested negative. So this is, like a 4-month period where he didn't have the virus, and then suddenly he had the virus. So of course, it offers some strong evidence there that he was reinfected with the same virus. But was he? Was he infected with the same virus? And that's where things get a little more interesting, not that things weren't already interesting. So what these researchers did is they tested the virus from back in March, because they had samples from March, and they tested that virus, the genome of that virus, so the genetics of that virus, against the genome of this new virus that he was reinfected with. And they wanted to know, what are the differences between these two viruses? Are they exactly the same, or are they a little bit different, or are they different in key ways? So they did a whole genome analysis, which is something that you can do really for any number of different things, but certainly for viruses, it's pretty simple. And they again confirmed that it was not a resurgence of that first virus, because there were key differences between the second infection and the first infection. He was, in fact, reinfected according to antibody response. So he had an antibody response again, and according to the viral load, which is what I mentioned earlier. So the differences were, and again, this is something that if you follow me on Instagram, I'm going to be posting this, so you'll be able to actually see this in detail if you're interested. But you can look at a really simple diagram of the genome, so the gene sequence that makes up the virus, and it's made up of 2 sections known as an open reading frame, which is where you express the particular genes that make up that genome. And I'm going to focus on one particular section in just a second, but overall, so by comparison of the 2 viruses, there were 23 nucleotide differences. Meaning, what is a nucleotide? So nucleotide is a reading segment of a gene. So for a gene to be read, just like letters are on a page, for a word to be read, you need the letters, right? In a correct organization. So the word is WORD. So you need to have those 4 letters in that organization so that you can read in your head, oh, that's word. Okay. Same thing with a gene. So if you have a gene of 4 nucleotides, so the nucleotides would represent 1 letter, then you're able to read that gene and it's going to tell you something. It's going to tell the cell something, and it's going to be able to produce a particular molecule out, or a particular protein specifically out of that gene. However, the differences in these 2 viral genomes is 23 nucleotide differences. So 23 of those letters had been switched, had been changed, and that is caused by mutations. And this happens especially with RNA-based viruses, so ribonucleic acid viruses. These are highly mutagenic viruses that mutate quite a lot, so they make a lot of different changes. It's not like they're consciously thinking about it. It just happens spontaneously. If that's sunlight that's interacting with it, if that's reactive oxygen species, if that's something else that interacts with those nucleotides and just changes the chemical structure just a little bit to end up allowing for this mutation to occur. So 23 changes, 23 mutations occurred in the second virus once he was reinfected. But here's the kicker, that only 13 of them were what's called non-synonymous, meaning that sometimes, which isn't exactly akin to like words, but if you were to change one of these nucleotides with another nucleotide, sometimes the gene gets read the exact same way. So, and that's where I'll leave that. That's what's called synonymous. So you get this shift in, let's say, an A to, or from, let's say, an O, if we're going back to our word reference, from an O to an A. But your brain kind of reads it anyways as word, as the entirety as word. So that's can also happen in genes. And so 23 nucleotide differences, but 13 of them were non-synonymous, meaning that there were mutations or changes in the nucleotides that led to a differential expression of that gene. So now the gene is no longer expressing the same thing, but it's actually expressing something else, some other protein or another amino acid in this situation. So if we focus in on one section, which is one of the primary sections that people have been really focused on when talking about this particular coronavirus, is the spike protein. So the spike protein is, if you've seen images, and again, if you want to actually look at the images, you can hop on over to my Instagram. The protein is kind of a ball, right? And then it has these little things, these protrusions that stick out, and they're all over the ball. Those are the spike proteins. And the spike proteins, because they look like spikes, the spike proteins are the parts that actually attach to the cell. And we know that that's the angiotensin converting enzyme receptor. So it binds to that, and then it gets taken up by the cell. So that spike protein in this second virus, the second time he was infected, had 4 of these non-synonymous mutations. And those are the ones that you're most interested in. So out of the 23 nucleotides, 13 of them were non-synonymous. But in the spike protein, there were 4 changes. So 4 of these led to 4 different amino acids that were ended up being changed. So that leads to, if you were then to compare the spike protein of the first virus to the spike protein of the second virus, again, just looking at this one individual, then you would actually see structural differences. The actual protein itself would be slightly different in its structure. Now, that could make it worse at infecting the cells, or it could make it better at infecting the cells, or it could make 0 difference whatsoever. You don't know until you actually run tests for this kind of stuff. But So focusing in on that spike, you get these 4 different amino acids that are different. But the great thing is, and there's good news and there's bad news. So I'll tell you the good news first. Let's try and stick to the optimistic part first. That he was asymptomatic or unsymptomatic the entire time that he was in the hospital. So again, he was picked up by the authorities. They said, okay, you have coronavirus, again, COVID-19. And he's probably like, well, I feel 100% fine. And I had it back in March and I went through hell during that time. So why don't I feel really crappy now? And he never displayed any sort of symptoms. So they did a bunch of blood tests. And again, they found no differences. I even told you about some of the markers, looking at some of the immune cells. None of those were elevated or decreased or anything like that. They did x-rays and they did different radiographs and whatnot, and they found no differences, no changes, no abnormalities. So this guy's completely unsymptomatic. So if they hadn't done that screening, he would have just gone through into society and continued to affect people. And he wouldn't have no idea until he finally recovered. So that's a positive, the fact that he didn't have symptoms, in terms of like, well, hopefully maybe in that case, if we can extrapolate that out, which is a huge if we can extrapolate that out, then maybe if a person gets reinfected by the virus, then maybe they'll have mild symptoms, which is great. Mild as in like they don't even feel it. They don't even know. However, if that is the case, that a person can be reinfected, then that also means that herd immunity probably isn't going to work. And if you've heard of that term before, herd immunity is the idea that if a certain percentage of the population gets infected and has the virus already, then they build up an immunity, and therefore then they don't have to worry about it. And it also protects the other people around you, because you have antibodies against and protective mechanisms against this virus. But here we're already getting proof 4 months later that people can be reinfected. So this also, unfortunately, also affects our vaccine research, because this also limits the length of the effectiveness of the vaccine, right? Because the vaccine is supposed to sort of allow you to experience the benefits of your immune response dealing with this virus without actually having to go through all the symptoms of the virus itself. So this suddenly cuts down the length of the effectiveness of the vaccine. Now, does this mean that the vaccine will be useless? Well, no, because of that very first piece of positive information that, and you could make that argument for herd immunity as well, but there is a problem with that I'll get to in just a second. That because you have this sort of unsymptomatic, really mild symptoms, that at least if you get a vaccine and you get an immune response, then the idea is that, again, if this translates to other people, that other people would also be non-symptomatic, then that'd be great. I mean, then sure, you might get the virus and still technically have the virus, but at least then you wouldn't know it. You wouldn't feel it. And if then it's pretty inert. It's not going to have much of an effect, or at least not a noticeable effect, I should say, because we don't know how this will affect people long term, many years down the road. Now, you could also make that argument for herd immunity, right? Well, okay, if a lot of people get it, then okay, well, we don't have to worry about it as much then, because if I get it again, it'll be mild. But we don't actually, this isn't a 33-year-old guy who's healthy, so largely in good health. So if that's the case, like, is that necessarily going to translate to somebody who's 69 years old, or maybe has pre-existing health conditions, or has diabetes, whatever it might be? We don't know that. This is simply a case study. This is only a proof that we can be reinfected by coronavirus. We don't even know if this happens on a large scale, if some people are immune for longer than other people. We just know that it is possible that we can be reinfected with this SARS-CoV-2 leading to COVID-19. So, And it also brings up questions of if a vaccine is if we're going to have one vaccine at one point and then another vaccine later on down the road and another one and so on and so forth, kind of making it turn out like the flu vaccine, like influenza, where you're kind of picking and choosing and hoping to pick up different flu vaccines, or in this case, different versions of SARS-CoV-2, and hoping that your immune system has enough of a response to the initial one that it will also attack some of the other ones. So the idea there is that T cells tend to target, T cells are cells that will attack, or are part of the adaptive immune system. So if they allow our body to have a much more detailed, specific response to a particular pathogen, in this case, SARS-CoV-2. So they would attack that particular molecule or particle, the virus itself, and they specifically target sections of that virus. So they identify, oh, I've seen this before, so I'm going to attack that. But like I said, that there were 13 differences in this virus. So the hope and the idea, what these researchers proposed, is that the T cells are still able to somewhat recognize the new virus, and that's why he didn't have any strong symptoms as a result. So in conclusion, this tells us for sure that we can be reinfected with coronavirus, or at least some people can, and probably a lot of people, if not everyone. This also tells us that we might have mild symptoms upon reinfection. Again, we would need many more people to be reinfected to actually know that for certain. It removes the idea of herd immunity, or at least the way that we were thinking about it, and it also limits the length of the vaccine. That doesn't mean that it limits the effectiveness of the vaccine, but it does at least provide some proof that will likely need to have multiple vaccines, or if that one vaccine has a good baseline, you can just hope that your immune system can still attack other versions that slightly deviate from the original version of that virus. All right. That's what I thought. This was really interesting. It certainly opens up, a Pandora's box in terms of what this means for the future. But regardless, we'll find a solution. And like I said, I don't think that the vaccines are suddenly null and void. They are going to be absolutely beneficial. It's just that they may have extra limitations to them that we were hoping may not exist. Okay, well, with that said, I hope this was informative for you, and I hope to have the absolute pleasure, as usual, to speak with you in the next podcast episode. Have a good one. See ya.